Alright, so this is going to be the walkthrough through our enzymes gizmo. Once you have launched it, you go ahead and hit start. You are a veterinary technician working at a small animal hospital. It is your job to help animals maintain good health and recover from injury or disease. Click here to see your first patient. We follow the blue. Your patient is a Great Dane called Claire. Claire's owner brought her to the hospital because Claire is eating a lot of food but is losing weight. Let's see her history. She is a Great Dane. She is 1 year old. Her weight is 45 pounds when it normally is 90. She is female. In her early life, there was no history of disease and her weight was 76 pounds. She now has had weight loss and lost more than 30 pounds while still eating normally. The other symptoms are that there is not enough energy to play and she has an increased appetite. Let's begin our investigation. Claire's symptoms suggest that there is a problem with her metabolism. If we click on the orange metabolism word, it will open up our glossary. Anytime you see a glossary term, you may open this up to get a definition. The way we navigate is through any of these blue blinking buttons. Metabolism is the term used for all of the chemical reactions that happen in an organism, such as the plant or an animal. A healthy metabolism is important in maintaining homeostasis. So Claire had an increased appetite, low energy, and she's eating, but there's no weight gain. Let's click to continue. To help restore homeostasis in Claire's body, you first need to learn more about metabolism. Let's enter our metabolism handbook. Welcome to the Enzymes Handbook. Here we are going to use the arrows to flip through the pages. What is metabolism? Metabolism is the term used for all of the chemical reactions that happen in the body. All the chemical reactions equal your metabolism. An organism's metabolism includes two types of reactions. One type is reactions that break things down. For example, chemical reactions are used to break down food. The second type is reactions that build things up. For example, chemical reactions are used to build muscles in the body. Let's look at chemical reactions in more detail. Take a look inside of the cell. Reactants and products. All chemical reactions involve chemical substances called reactants that are changed into different substances called products. Here are the two reactants. Let's see our chemical reaction. The two reactants come together and end up forming a new substance. In the chemical reaction below, we want to predict what will happen to the amount of reactants and products when the chemical reaction starts. Here, you will want to answer whether you think the reactants will decrease, have no change, or increase. Same thing with the products. Will they decrease, have no change, or increase? You will not be able to go past this without answering these two questions and hitting submit. Our reaction rate. The speed at which a chemical reaction happens is called the reaction rate. In science, the word rate refers to how quickly something changes over time. Our reaction time for 2 seconds. We will look at the number of products made in 2 seconds from a chemical reaction. First, we are going to click play to see how many products are made in 2 seconds when the reaction rate is slow. When it's the slow reaction rate, we make 4 products. Now let's try with the fast reaction rate. With the fast reaction rate, we make 6 products. Our reaction rate formula. For example, when this reaction was performed at the slow rate, 4 product molecules were made over 2 seconds. This would be a 2 products per second. Let's enter this data into the reaction rate formula. The amount of product divided by time equals our rate. When we enter the data, 4 products divided by 2 seconds equals 2 products per second. Graphs allow scientists to analyze our data. Let's see what this reaction looks like on a graph. On the y-axis, we will put the amount of product made. Next, we will show time in seconds on the x-axis. The reaction made 4 milligrams of product in 2 seconds. Let's first put the amount of product, 4 milligrams, on the y-axis. Next, we will add the time data of 2 seconds. Now we will click the intersection of the dotted lines to plot our data point. Now we started with 0 product at 0 seconds, so we'll plot 0, 0. And when we click the 2, it forms our line, and we can click the dots to connect the data points. Now here, using this graph, how much product would be made if we have stopped the reaction at 4 seconds? So here you would look at the graph, 1, 2, 3, 4, follow it up on the line, and then across to read this. Again, this is a question that you will have to answer and hit submit to move on. This is a graph for a different reaction. Here, the amount of product is measured in milligrams. A milligram is 1,000th of a gram. What is the reaction rate of this graph? Here, we can either look at our point where we have had 16 milligrams in 4 seconds, or because all of these are written in seconds, we can look at 1 second, go up to the line, and go across to where it reads 4. When you answer this question and hit submit, it will let you move on. Why are reaction rates important? For organisms to survive and grow, their chemical reactions must happen quickly. Let's add our enzyme. Chemical reactions break down food. Adding enzymes speed up digestion. Chemical reactions build muscles. Adding enzymes speed up muscle growth. Let's learn more about enzymes. What is an enzyme? An enzyme is a protein that makes a chemical reaction happen faster. Substances that increase the rate of a chemical reaction are called catalysts. Enzymes are the catalysts that organisms use to speed up their chemical reactions. For example, the reaction of nitrogen and hydrogen to form ammonia, NH3, is important in both organisms and in chemistry. This reaction is slow, and the rate must be increased with a catalyst. Enzymes catalyze this reaction in cells. In a cell, the catalyst is the enzyme. In chemistry labs, iron is used as the catalyst. In a lab, the catalyst is iron. Let's catalyze these reactions. They both made ammonia. Enzymes are a type of catalyst. All enzymes are a catalyst, but not all catalysts are enzymes. What effect would the enzyme have on the amount of product made in 3 seconds? Would it decrease, have no change, or increase? You want to answer this and hit submit, and you will get an additional question of what effect would the enzyme have on the time to make 1 milligram of product? You want to answer this one and hit submit here as well. The reaction rate of the chemical reaction was measured with and without an enzyme. Which line is the reaction with the enzyme? Here, you want to choose which one of these lines on the graph you think is the reaction with the enzyme. When you are justifying your answer, you want to answer these 2 questions. First, I want you to think about what does the enzyme do to a reaction? And then, what would that look like on the graph? When you answer those two questions, you will have a good justification. We can now hit Submit. Now we will test our predictions with an experiment. Let's test our predictions with an experiment. The two cells below contain the reactants needed for the ammonia reaction. We will add enzymes to one of the cells. Let's start our reaction. A lot more products were made with the enzymes. Now we can click here and return to the questions, and here you'll be able to see whether or not you had those answers correct. There are over 75,000 different enzymes in the body, and each one has a specific job. Enzymes are a type of protein, and like other proteins, they are made of small building blocks called amino acids. Every enzyme has a different sequence or order of amino acids, so every enzyme has a different structure or shape. Every enzyme has an area called the active site. This is where the reactants are changed into products. Each enzyme's active site has a different shape. You can think of this as being like a lock. Just like you need a specific key to fit into a lock, only the reactants with a specific shape can fit into an enzyme's active site. Reactants that bind or attach in an enzyme are also called substrates. Here is a question that you will need to answer to move on. Which of these molecules is the substrate for this enzyme? Once the substrates are bound or attached, the enzyme catalyzes the reaction and converts the substrates into products. Notice some of the enzymes make a molecule and some of the enzymes break a molecule. Here is another question that you will want to answer to move on. Which of the following is false about the structure of enzymes? The shape of an enzyme also depends on temperature and pH. Even a slight temperature and pH change can change the shape of the enzyme. This shape change can stop the enzyme from working properly. Let's see what happens when we increase our temperature. Our shape changed and the substrate will not fit the active site. Let's reset, and we can see that the substrate now is able to fit and the reaction occurs. Now let's see what happens when we decrease our temperature. Notice the shape of this enzyme changed, but in a different way, but it still caused the substrate to not fit the active site. Now let's look at the effects of pH. Let's see what happens to this enzyme when the pH increases. Notice that we had a denaturing and the substrate will not fit the active site. Let's now check when we decrease the pH. Again, it is a different shape, but the substrate still does not fit the active site. Now that we have learned about enzymes and metabolism, we can return to the case to investigate Claire's metabolism problems. Remember, enzymes are catalysts. They speed up our chemical reactions by lowering the activation energy. Enzymes are specific. The active sites can only catalyze specific reactions with specific substrates, like a lock and key. And enzymes are sensitive. Slight changes in pH or temperature can cause an enzyme to stop working. Now we will return back to the case.